The tropical forests in the western Andes of Ecuador lie along a steep elevation gradient that stretches from 700 to 3,500 meters. The abrupt changes in elevation across this gradient creates different conditions of temperature, humidity, and air pressure, which in turn gives opportunity to a myriad of organisms to adapt to these changing conditions. Hummingbirds, for example, have evolved into dozens of species with particular adaptations to environmental conditions, but also to the flowers they rely on. Hummingbirds are exclusively found in the Americas, and this in the northwestern portion of the Andes, where this group reaches its highest diversity. Up to 40 species could coexist in a single site. The plants that hummingbirds use as nectar source are also greatly diverse in this region. More than 400 species of plants are known to be visited by hummingbirds. Thus, interactions among plants and hummingbirds sustain an important ecosystem service in the Andes. Pollination.
The Andean region has been populated by humans for centuries. The interplay between human activity and biophysical conditions of the Andes has created a mosaic of landscapes and land use regimes, in which the remaining Andean forests play a critical role for the maintaining of biological diversity and ecological processes. Land use change from forest to pasture is a common practice in our study region. Cattle growth activity has resulted in a different landscape configuration and different levels of land use intensity. How land use change affect the diversity of plants and hummingbirds and the way they interact is the central question of our research. For this, we integrate different field and analytical approaches to document plant-hummingbird interactions along gradients of land use. First, we quantify how much resource is available both in the forest and deforested sites. For this, we perform bi-monthly census of open flowers along 14 transects of 1.5 kilometer length. We select flowers to count on the basis of the so-called ornithophilus syndrome, which means that plants have bright-colored tubular flowers. In front of each of these flowers, we set time-lapse cameras that take pictures per second, so we can record which species of hummingbirds are visiting which flowers. Videos obtained are analyzed with a deep learning procedure which select interaction candidate frames. Then we review those frames and retrieve the information about the interactions. Ultimately, we aim to learn which species of plants and hummingbirds are lost during deforestation and how these local extinctions influence plant-hummingbird interaction networks.
Information obtained through this project has been critical for the design of conservation actions. For instance, we know now which species of plants are important in the diet of the black-breasted pablet, a hummingbird highly threatened and endemic of Ecuador. Future habitat restoration efforts will focus on these plants, but also on plants that are important for other hummingbirds as well.